somebody I've met. I think the first time we met was in Columbia. We overlapped in visiting Columbia and we were giving seminars the same day.
Yeah.
Yeah, that was probably the first time. And the last time was in Amsterdam. So, Professor Backx, Peter Backx, is coming to give our seminar from Toronto, and I was very happy that he made it yesterday because of all the flight delays, because of those major snowstorms from the East Coast that apparently didn't affect Toronto much. And so he's in Toronto now, and before Toronto, where he's a professor in physiology and medicine, and also a researcher in the Healthcare Research Institute at Toronto Hospital, and has done some really groundbreaking work and beautiful work on ion channels in the heart, structure and function of ion channels in the heart. He is a veterinarian by training, and then did his Ph.D. in Calgary. It's a beautiful part of the world, with Hector Kurz. From there to Eduardo Marban's laboratory at Johns Hopkins, where at the time there a lot of work was done on ion channels in the cardiovascular system. And then Toronto, or Canada in general.
Yeah.
And without further ado, thank you for coming.
Thank you very much. So thanks very much for the invitation. I'm honored to be here. Hopefully my seminar will be of some interest to many of you, and I hope to convince you. I've changed my title a little bit, but what I'm going to end on is the notion that atrial fibrillation or atrial remodeling that I'm going to show you happens in exercise mice is not unanticipated from the human studies, and probably connects up to AF, atrial fibrillation. I'll probably refer to that as AF for the remainder of the talk, is without question related, right? Or at least in my mind it's related. We'll see. We'll see if I can convince you. The two people who actually did all the work I'm going to talk about are a very talented postdoc, Ruz Sobi, and a master's student, Farzad Izzaduzdar, right? I'm reluctant to pronounce his name. Anyway, those are two wonderful guys I had the opportunity to work with, or who worked with me. And then down here are less important people who we collaborated with. This guy happens to be an EP cardiologist, as he, and they were involved in doing my ablation actually a few years ago. The reason that I'm interested in atrial fibrillation is because I have it. I suffered from it for 10 years, managed it, and there's little doubt in my mind that there's a connection between my AF, and a number of people, and exercise. And this sounds a little bit like heresy, right? Exercise is very, very good for the cardiovascular system. There's no question it's good for the heart. And so, you know, if you go through the literature and even the common, you know, on the web, television, the common media, you're going to hear lots about how good exercise actually is, and it's not only good for the heart, but it's actually good for the entire body, right? And yet, I'm going to try to convince you that too much of a good thing actually does have a little bit of downside. I'm not entertaining, and I'm not going to promote the idea that you shouldn't go out and exercise. That's the last message I want to leave you. But there are consequences of, I would say, excessive exercise, and I'll try to make that clear in terms of what that means. And here's just a little bit of evidence for, you know, how important exercise actually is. And in fact, if you don't exercise, you have much more greater issues with cardiovascular health than if you don't exercise. And that's very well summarized by this graph here, by Myers, which shows relative risk of death, many of these actually being cardiovascular related, as a function of how much you actually exercise. And the most fit people clearly have by far the least number of adverse cardiovascular events. And that's in a healthy population. If you actually look in patients that have coronary artery disease, same thing. If you actually ask the question, you know, what's your likelihood of actually having a cardiovascular event, the more you exercise, right, is the more protected you actually are. So both in healthy people and in individuals that have cardiovascular disease, there's little question that exercise is actually a very, very potent protector of cardiovascular disease. But over the last probably 10 years or so, there's been suggestions that this is a U-shaped relationship. So if you plot sort of physical activity, and the way this is done is in terms of metabolic equivalence, right? So the baseline level of activity is one METs. And so as you increase your number of METs, and this would be, really, really high-end activity associated possibly with, maximal exertion, there's a suggestion that if you do that over long periods of time, over many years, that there's actually lots of potential adverse effects. And some examples of that are shown here. So, so the first thing is that you've got, for sure, there's no question that, if you've got an underlying genetic disorder, sudden cardiac death can be brought on by exercise, often vigorous exercise. And certainly in coronary artery disease patients, there's also an increased risk of you actually having a coronary event. And this is actually shown in this particular slide, this concept, in individuals that actually have cardiovascular disease. And if you actually increase your level of activity per week to about 50 times your sort of baseline metabolic rate, there is clear evidence for an increase in mortality in those particular individuals. So there are vulnerable populations, right? And here's an example of individuals that have right ventricular enlargement, or rather right ventricular dysplasia, arrhythmogenic right ventricular dysplasia. And as you increase the level of exercise in those individuals, they also have an increase in adverse effects. And associated with this, in normal populations, if you actually look at it, it's very hard to see here, and I tried to reproduce it accurately from this particular paper, but this is actually showing levels of troponin release, troponin I in the blood. And in fact, if you look at the levels with high exertion, they actually rival the levels that you actually get with a coronary event, right, with myocardial infarction. So it's pretty clear that there's a number of different conditions that actually lead to adverse effects. I won't dwell on this notion, and I'll just leave off the others, which are associated with veteran athletes in the wrong direction here. Okay, so what about AF? So that's just sort of general cardiovascular disease. And if you ask the question, how does exercise relate to AF? Well, first of all, I just want to remind you of who actually gets AF, and that's just illustrated here. And there's really two features, right, of things that drive atrial fibrillation. The first is an underlying cardiovascular conditions, and the second is age, right? As you increase in age, the likelihood of you developing atrial fibrillation goes up dramatically, and that's exacerbated if you actually have various types of cardiovascular conditions, and in particular hypertension. But many of these, I'll point out here, are associated with conditions often that actually involve low levels of exercise, right? So relative inactivity, things like diabetes, metabolic disorders have been actually linked. And there have been a number of clinical trials that have actually looked at the impact of exercise on atrial fibrillation. And the majority of these have actually demonstrated in relatively unfit individuals that if you increase your level of, these are the highest or the most fit individuals who could actually exert themselves to greater than 11 METs on an exertion test, had a much less likely, were much less likely to develop atrial fibrillation over a period of a couple of years versus those that were the least fit. And if you actually take individuals and put them into exercise programs, you get essentially the same observation. The more fit you are, is the less likely you are to actually develop atrial fibrillation. So this is, again, the most fit individuals over a period of time, and this is the question of whether or not they remain AF-free or not. So it's pretty clear that with AF as well, there's, or at least atrial health, there's clear benefits actually of exercise. But there's a few studies that came out over the years and actually nicely summarized in a paper by Mont in 2009. And this is in the midst of when I was dealing with my AF. And I used to be a competitive soccer player. And this summarized a bunch of small studies that showed that your odds ratio of actually developing atrial fibrillation in an otherwise relatively normal population without any evidence of cardiovascular disease was 5 times greater than individuals that were sort of matched up. And this paper actually, or this summary paper, actually drew a lot of attention and a lot of criticism. And then in 2013, there was a study that was done coming out of Sweden where they looked at 52,000 cross-country skiers, and they showed very clearly that that not only was there a connection between exercise, but those individuals that actually had the best finishing times and those that actually ran the most races had by far a much higher odds ratio of actually developing atrial fibrillation. Interestingly enough, both of these sets, all these studies in this huge study, all were done in relatively aging athletes, right? And if you talk to cardiologists who deal with atrial fibrillation, they'll tell you that, of course, it's, as I pointed out, age is a major confounding factor in all of this. But it's interesting to note that if you actually look at, there's many examples actually of athletes, and I've just taken 5 of them here, who actually develop atrial fibrillation at a young age. And it tends to be in highly competitive athletes, usually dealing with, in sports that are considered endurance sports, right, that require relatively high cardiac output. In the NHL, which of course in Canada is like, it's like your football, I guess, right? Virtually every NHL team has 1 or 2 players that are actually dealing, and these are young men that are actually dealing with atrial PACs, and many of them develop atrial fibrillation. Not many of the players, but a percentage of them on every team. So it's pretty clear, I think, from the literature that there's this U-shaped dependence that I talked about in cardiovascular health also appears to apply to atrial fibrillation. So in other words, if you take the most sedentary individuals as sort of your controls, if you actually increase your level of exercise light to moderate, even to high levels, you actually reduce the likelihood of you developing atrial fibrillation or adverse atrial remodeling. But if you look at the endurance sports, and those individuals that are relatively engaged in sort of obsessive, almost sports activity, their odds ratio of developing AF actually rivals that of hypertension and diabetes. Okay, and so this again, this is a very nice study that just came out that actually did true controls. So they actually got rid of all the other confounding factors and compared individuals that had the same body mass, etc. The only difference was their level of activity. And you can see that there's a clear, relatively strong dependence, which was what Mont had actually pointed out. I just want to emphasize that this is individuals almost invariably that are involved in endurance sports. So what are those? Well, many of those are things like hockey, soccer, biathlons, cross-country skiing, rowing, cycling is another large one. And most of the time these individuals are what are called lone AF patients, which just refers to the fact that they have no other cardiovascular disease. And it's very common for them to develop their AF in response to meals, sleep, or exercise. The standard sort of the profile of these individuals is usually they're in the early 40s and up in many cases, but relatively young compared to those individuals that are actually developing AF when they have coexisting cardiovascular disease. And Mont estimates in this review article that about 60% of the lone AF patients that show up at a clinic are engaged in some excessive sporting activity. Whether that's an accurate number, I don't know, but that was the number he actually suggested. And I'm going to come back to this, but one of the common features of these individuals is that they have elevated vagal tone in response to the exercise. They often have enlarged atria as well as ventricles. And the reality is that nobody really knows what causes this, right? These are modulating factors for sure, but no one really understands entirely what's actually driving atrial fibrillation in this population. And I'm going to come back and try to convince you that it's actually probably the same kind of mechanisms that you're actually picking up in AF driven by other cardiovascular conditions. And there's the other factor I just want to draw your attention to is that this is still a relatively small fraction of the population. So it's very clear, or it's very likely, that there are underlying genetic factors. Every person that engages in high-level endurance sport doesn't get AF, right? It's only a subset of these individuals likely have some sort of vulnerability. No one's actually worked out those details as of yet. Now, here again is my disclaimer, right? So if I've given this talk now about 3 or 4 times, and at the end of every talk, someone will put up their hand and say, well, you've just given me a reason not to go out and do exercise. And so I've actually put this slide in to discourage that kind of interpretation, because if you actually look at endurance athletes and you ask the question of what their survival is, and there's about 4 or 5 studies that have done this on a few different continents. And it's very clear, in this particular case it was in cyclists, and it's very clear that even though they may have an increased vulnerability to AF, they actually live longer, right? So it's a bit of a pain it's a bit of a pain to have to deal with if you've had AF, but the bottom line is you actually will live longer regardless. So I was very interested once I developed AF to try to understand why I might be developing AF, and so I actually shifted some of my research. That's the beauty of being in an academic center. You can actually wake up one day and say, hey, you know, I'm going to go chase this down for a little while. And we've had a lot of fun actually doing it. I've actually always been a sports buff, and so I've learned a lot about sort of exercise physiology along the way. So it's really been a good ride, and fortunately had some good people to do the study. So here was our initial objective, right, was to develop mouse models of exercise. It wasn't that simple, actually, but we thought it would be quite simple. And then test for a connection with AF. So the first 2 models that we worked on are shown here. I'm sorry for the background noise here. Treadmill running and swing exercise. And this isn't really accurate, because the way we do our swing is we actually, let me just shut this off, it's a little annoying, is we actually run a current. And we found that that was actually imperative to get them to actually swim. And I'll tell you a little bit more about what their METs were a little bit later. But the level at which they're exercising is comparable to, you know, sort of endurance athletes, although it has to be scaled by mouse to man, of course. So here's just some characteristics of what happens. Now, I should mention that these training periods were for 6 weeks, right? And so this is most of the data I'm going to show you is after 6 weeks. We did some other interventions as well, but most of it is that. So here's what happens to the mean heart rate over time. Very similar to what you see in humans, right? There's a reduction in heart rate, right, as you train and as you get more fit. And this happens actually relatively quickly. Within 6 weeks, you can see more or less stabilizing at under 500 beats per minute. And it's not dramatic, but it's very consistent finding. And if you look at the circadian rhythm, in fact, it's day and night. You see this? When you take the hearts out, in fact, they're a little bit larger, which is what's known from human literature, and that's just tabulated here, right? So the hearts are bigger, and in fact, the hearts are a little bit stronger. If you actually look at dPdt max, which is a measure of the speed with which, or the rate at which the hearts can actually develop pressure, dramatically elevated actually, as is the maximal responses to valbutamine, right? So these are clearly strengthened hearts, which is sort of typical of what you'd expect, right, with exercised hearts. And I'll say no more about that. So more or less what we expected. So the first question, of course, that we went to answer or determine was whether or not they developed atrial fibrillation. Now, these mice don't develop triggers. The normal AF involves, and I'll talk a little bit more about this in a minute, involves some triggers from some part of the atrium that then drive abnormal electrical activity, and so you've got this concept of trigger and substrate. We don't have triggers in our mice as far as we can tell. Like when we've done implanted ECG measurements, telemetry, we could never really pick up any triggers. So the way we do our experiments to interrogate whether or not they are have atrial remodeling is sticking a catheter down into the right ventricle and the right atrium. and then do some overdrive stimulation, and that's actually shown here in sedentaries. And you can get these to actually drive into some sort of non-sustained arrhythmia. But if you compare the swim versus the treadmills, running versus the sedentaries, a couple things. One is you actually get quite different, often very different behavior with programmed stimulation, and the durations of these events are actually considerably longer following exercise versus sedentary controls. And this just shows you sort of the frequency with which one would actually pick up these arrhythmias, which were defined as anything that's longer than 10 seconds in these mice. And you can see about half of them, right, between 40 and 50% of these animals, whether they're swimming or treadmill, you can actually pick up relatively prolonged atrial activity. Now, in the ventricle, on the other hand, actually there was tended to be a reduction. So whatever's going on with exercise, it's unique to the atria, and the ventricles are actually, if anything, are protected. And I just threw this in to point out that, and I mentioned this to Yarn last night, that if you actually look at the patterns that you pick up on surface ECG as well as with optical mapping, it's very complex, right? It's not an atrial tachycardia. It often goes, periodically into ATAC, but also often shows lots of fractionation and lots of complexity and randomness associated with those particular signals. So we went ahead and tried to understand what's going on. Like, why is there the difference between this exercise and these sedentaries? And of course, we know, right, what happens with atrial fibrillation is that you end up with sort of random electrical activity within the atria that then feed down onto the ventricles, and the net result is that you get this regular irregularity, right, to the QRS intervals, as opposed to normally where there's a nice clear connection between P wave, QRS, and repolarization. And this leads to lots of palpitations, reduced cardiac output, often reduced pressures. It can drive cardiomyopathy, and of course the primary problem, or a primary problem with AF, is the increased incidence of stroke. So that's just sort of a background for AF. I don't think I probably needed to mention that. But here's how we think about it, and here for the purposes of sort of dissecting out how we determined what was going on in our mice. I just introduced this concept, right? This idea that any abnormal electrical activity I mentioned already involves triggers and changes in automaticity, and very often re-entry type activity. And in order to get this kind of re-entry arrhythmia, the concept is that you need an excitable gap, right? And you need this period of refractoriness to be short enough that you can have an electrical activation that actually finds its way through the atrium or through the myocardium and rediscovers a region of that's excitable. And under those conditions, you can actually have the opportunity to develop abnormal rhythm. And the concept here is that this concept of wavelength, right? And the wavelength is essentially the effective refractory period, or the action potential duration, multiplied by the conduction velocity. And if you multiply those two out, this is a time parameter, right? This is a space over time. So you multiply those two out, and you get a length. Okay? And that's why it's called the wavelength. And when that wavelength conceptually is more or less than the size of your atrial tissue, you have the conditions that promote the possibility of atrial arrhythmias, or any arrhythmia for that matter. So that's sort of how I'm going to think about and demonstrate and show you how we thought about dissecting out these mice. Now, if you, in terms of atrial fibrillation, we know there are essentially three factors that contribute. I mentioned before the notion of triggers, right? Something has to get you into this situation where you've got the cat chasing its tail. And that often is set up by triggers. Once you have triggers, there's a couple factors that actually can influence the likelihood of you developing these reentrant type or these abnormal electrical patterns. One are modulators, such as the autonomic nervous system, in particular the vagus, right? And I'll come back to this, but this system actually will change the refractoriness, right? Also changes the intrinsic heart rate, and both of those can contribute, along with dispersion of repolarization to increasing the likelihood of you actually developing atrial fibrillation. Another modulator is actually atrial stretch and dilatation. Where I'm not going to actually come back to this. And these are modulators that if you have a substrate, you're also much more likely to develop, again, atrial fibrillation. And those substrate factors are things like atrial fibrosis, which we know is related to atrial fibrillation, and of course alterations in the EP parameters. So I'm going to just walk through from here and show you how we sort of tried to dissect out these different factors. So the first question is the modulators. So I already mentioned about the heart rate actually being reduced in our mice, typical of athletes, and that's again shown here. And we know what that originates from, right, at least in athletes, and we demonstrated it's the same in mice. There's actually an elevation in parasympathetic activity, which actually through the release of acetylcholine, right, through G protein coupled receptor, activates a potassium conductance called IKACH. And this is actually one of the primary factors, right, that regulates heart rate. And if you actually increase the level of vagal tone, you will actually slow the speed of with which, or the number of times that the SA node actually fires an action potential per unit time. And that's what you're actually seeing here, right? And if you actually apply atropine in order to block these receptors here, you find that you can actually elevate the heart rate in the exercise animals as well as in the sedentary animals, right? And it actually doesn't normalize them. And if you sort of break it down further, it turns out they actually not only have an elevation of parasympathetic activity, but they have a reduced sympathetic activity. But the bottom line here is that what I'm telling you is that there is an increase in vagal activity, which is responsible for the reduction in heart rate. Now, the reason that that's important is because of this. One of the things about the KACH current in the atrium is that it actually abbreviates the action potential. And remember, I told you that the wavelength is a product of the action potential duration, the conduction, and the conduction velocity. If you reduce the action potential duration, you will reduce the wavelength. And if that becomes markedly less than the atrial size, you have the conditions for an arrhythmia. And so this is actually just pointing out that this is relatively unique to the atrium. So these are atrial cardiomyocytes. If you apply acetylcholine, if you actually apply that to ventricular cells, you get a much more modest impact of the application. The reason is there's lots of muscarinic receptors. These receptors are abundant in the atria. Okay, so when we actually now applied these agents, right, to the animals, what we found was that there was actually a change in the effective refractoriness. So just like I showed you, the heart rates increased. In fact, the action potential durations also increased. And that would actually tend to prolong or increase the wavelength and actually reduce the likelihood of AF events. And that's indeed what we actually observed. Right? There's actually a reduction in the duration, and actually the number of animals that actually developed AF was also actually reduced. Now, we went ahead and actually dissected what was potentially responsible for the maintenance of AF, despite the fact that we've actually normalized or eliminated this difference in vagal tone. I won't go into the details of it, but it does turn out that if you look at the action potentials from the individual cells within the atria of the heart, when you isolate them, their APs are actually prolonged. They're not shortened, right? Whereas if you look in the intact animal and you ask the question, what's the effective refractory period? There's really no difference between the sedentaries and the exercise. Okay, so what's happening here is that the vagus is shortening up the AP, and if you didn't have the vagus, in fact, you'd have a prolongation of the action potential duration. And there's no difference in the actual sensitivity to acetylcholine. But the other thing we picked up actually was an increase in calcium current, which probably explains the prolongation of action potential duration. But the bottom line is that I'm just showing you this modulation, and I want to focus on the idea that we didn't get rid of all of the AF. And so that suggested to us that there's something fundamental in the substrate within the atria themselves that's actually been altered as a result of exercise. So what we did was we went ahead and we actually isolated atria. something like this, pinned them down, and then applied did optical mapping, right, with di4 and EPS. And here's what happens in a normal sinus rhythm in an isolated atrium. And here's where the SA node is approximately. You can see propagation clearly, and it's in sinus, and it's in sinus rhythm. Here's what happens in, now it's highly variable, right? But here's an example of what happens when you actually induce AF. Again, overdrive pace, and you can get lots of rotors, lots of heart block. This one happens to show very, very clear rotor activity. Not all of them did, but many of them did. Probably about half actually showed that kind of behavior. And I showed you before that about 50% of the mice develop AF. About the same number of atria, isolated atria, also underwent this abnormal pattern of activity, whereas very few of the atria isolated from sedentary mice actually did this. I guess I'm not really going to dwell on this. I threw this in because it's like bringing coal to Newcastle here, trying to tell people about arrhythmias at Wash U. But I just wanted to point out how complex these different patterns are. And if you look at, so the dominant frequency analysis, or if you look at the frequency of stimulation over different regions of these atria, you find it's highly dynamic. So different regions are going to, or the atria are going to show different patterns of excitability depending on when you actually do the analysis. So coming back to the point that these are highly complex arrhythmias, similar to what you actually might see, and certainly has been reported in sheep and dogs. And we analyzed it further to demonstrate that there's heart block and block conduction, etc. And again, I won't dwell on it, but again, just making the point that when you actually map these arrhythmias, they're actually highly complex, showing rotors that come and go, lots of conduction block, and disappearance of signals for periods, and then it comes back, etc. So it's pretty clear, right? Isolated atria, in the absence of any autonomic modulators, you can develop or you can induce atrial fibrillation in them. And this is just to demonstrate that one of the factors that might be at play here is just atrial size. I already showed you that the hearts are larger. If you actually look at the atrial weight to ventricular weight ratios, that should actually be VW, they're actually enlarged. So the atria are differentially increased in size, right, relative to the ventricles are larger, but the atria are even larger, right, for enlarged ventricles. And there's also prolongation of the P waves that occurs, and totally consistent with the notion that there's an increase in atrial size. So this is a potential contributing factor, right? Because the larger the atria is the more likely you are to be greater than the wavelength, the more likely you are to actually have an arrhythmia. So that's one factor. The second thing is we actually noticed in doing our conduction, our optical mapping, that our conduction velocities were quite a bit slower, actually, in the exercised animals. And that's just demonstrated here, and here's some isochrome maps, and I already mentioned this notion of the P wave. But when you actually look at connexins, there's no difference, right? These connexins are these proteins that connect one cell to another, and of course, they thereby influence conduction velocity, gap junctions here. But the more profound thing that we noted, or the large change that we noted, was a large amount of fibrosis that actually existed in the atria of the swim exercise mice compared to the sedentaries. And if anything, the amount of fibrosis in the amount of fibrous tissue in the ventricle, left ventricle, is actually reduced. So whatever's going on in exercise, right? There's differential growth of the increases in size, P wave prolongation. But there's also atrial-specific fibrosis that's actually occurring. So this is interesting, right? Because if you take atria from individuals that are in persistent AF, or individuals, where they're vulnerable to AF, one of the common features is actually fibrosis, right? And that's going to, again, slow conduction, decrease your lambda, increase the likelihood of you actually being able to develop. So the next thing that Farzad and Ruse did was we wanted to know what the molecular mechanism was, and so what we did initially was did microarrays. What was something we didn't expect, of course, metabolism lit up, right? So we did gene set enrichment analysis and gene ontology, and we used the biocarda pathways. And the things that lit up for us were metabolism. That shouldn't be too surprising. But the other one was this NF-kappa B TNF-alpha. And we were actually beside a lab of Stefan Bowles, who's a smooth muscle physiologist, or he's a vascular physiologist. And he has this data to suggest that TNF-alpha is actually a mechanosensor in the myogenic response. Like, I'm not going to get into the details of that, but that's a system that's designed to protect your capillaries, right, and ensure that capillary pressures are always at the levels that they actually should be. And he showed that if you interfered with TNF-alpha, he completely eliminates the myogenic response, a mechanosensitive transduction cascade. So we considered the possibility that that might actually be what's going on. He had the knockout mice, so we went ahead and actually just performed the experiment of knocking out TNF-alpha, or taking mice that had TNF-alpha. And here's what TNF-alpha does, right? First of all, it's a membrane-bound ligand that gets cleaved by TACE. It's released typically, although there's data to suggest that there's alternate signaling of these. It doesn't involve enzymatic activity. And it then binds to either, there's two receptors, TNF-alpha 1 and 2 receptor. And the TNF-alpha 1 receptor actually signals down through NF-kappa B through a relatively complex pathway, leading then to changes in transcription through NF-kappa B. And there's also clear evidence for activation of P38 simultaneously with TNF-alpha activation in many cell types. And this was interesting to us because P38 is actually known in fibroblasts, right, to be a major factor for SMAD signaling and leading to driving the collagen expression patterns in fibroblasts. So the first thing that Ruse and as far as I did was they just tried to find evidence for activation of TNF-alpha and NF-kappa B and P38. The first thing is that if you look at I-kappa B, which actually is an upstream regulator, right, that ultimately leads to the release of NF-kappa B. In fact, the level of phosphorylation is increased, and this is consistent with increased TNF-alpha signaling. P38 phosphorylation was also increased, right? So this to us actually suggested, right, that there was, activation of this particular pathway. And this was done after only 30 minutes or 4 sessions of actually swim exercise. And I won't get into the other details, but there's clear evidence for activation. And TNF-alpha is actually an inflammatory factor, right? It's been described primarily in macrophages. And that was interesting to us as well once we saw this, because the other feature of atrial fibrillation clinically in humans is that atria often have a lot of inflammation. So they have fibrosis and inflammation. This is a factor that actually ties those two together potentially. So we did the obvious experiment I mentioned already, TNF-alpha knockout mice we got from Dr. Bowles. We used the Tanacept, which is a recombinant scavenger of TNF-alpha. It's used quite extensively in people with rheumatoid arthritis. It's actually tried at one time in heart failure because it's a highly inflammatory condition as well. And so we treated our mice with this human form of the Tanacept, as well as a P38 inhibitor. And what's very interesting is that all 3 of those treatments completely eliminated the susceptibility to atrial fibrillation. So this is the control data, and you can see a Tanacept or the knockout or the treatment with P38 all eliminated completely, not only the AF, but remarkably, the fibrosis was completely prevented by these particular treatments. And yet, if you actually take a look at these mice and ask the question, all this other kind of remodeling that happens with exercise, does that still occur? And the answer is yes. So they still get enlargement of the atria, they still get enlargement of the ventricles, they still have increased contractility, they still get changes in their ERP, etc., right? And yet they don't develop fibrosis any longer, or we can't induce atrial fibrillation. So I'm going to make a summary, and then I'm going to just tell you the stuff we've been doing since, because most of this, almost everything that I showed you above has actually already been published. So what I showed you is that, first of all, I made the argument that endurance exercise is linked to AF. If you intensely swim or intensely run mice, they will increase their vulnerability to AF and inducibility, and they develop fibrosis as well as inflammation. I sort of stepped over that part, but there's elevations in macrophage numbers within the tissues themselves. We know that TNF alpha, NF kappa B, P38 are implicated, right? Because if you interfere with those pathways, we could prevent the fibrosis, and I think that's the most profound thing that we actually found. But this mechanism is not known, right? We still don't really know what the mechanism is. We found a player, right, in the transduction cascade, but we really don't know what's going on in terms of what started this. And I come back to the point I made earlier about biomechanical stress. So this is taken from a review article. Unfortunately, I forgot to put the author here. But the point is that biomechanical stress has been shown as an alternate form of signaling through TNF alpha. It actually doesn't involve cleavage of the TNF alpha through TACE. It somehow involves direct interactions of the TNF alpha ligand with its receptor. So that concept is actually out there. So here's some data that we generated to support the notion that this could actually be, at playing a role in our system. So we actually did telemetry recordings where we put pressure transducers into the mice, and then we exercised them. And here's what happens during an exercise bout. So this is time 0, and at this time the mouse is actually thrown into the tub to swim. And by the way, the mice actually, they're CD1 mice, I forgot to mention that. But after their initial training period, the mice actually love their daily swims. Right? They literally, if you put them on their hands, they don't run away. They actually almost jump off your hands. They're just like people. Right? Some of them love their exercise, and most of them do in the case of CD1s. Not true of all strains, by the way, and that's an interesting aside. Now, if you ask the question, what happens in response to throwing them in the water in terms of their blood pressures? Well, their blood pressures go way up to 160 and settle back down at about 130, and they start at around 120, right? This is exactly what happens in athletes, right? And it's known that you have to jack up your systolic blood pressure, right, to be able to perform as an athlete, right? This is an essential part of the exercise response. But here's the other thing that happens. If you look on the back side of the heart, there's a large increase, right, in the amount of pressure that's on the back side of the heart, right? So the filling pressure that the heart actually sees goes up. And that's a well-known aspect of the Starling mechanism. It's how you increase, one of the ways you increase cardiac output. In humans, there's lots of studies that have shown these go from about 2 or 3 millimeters of mercury to 20 to 30. If you actually go to very, very vigorous exercise, it goes well above 30 millimeters of mercury. That's a huge, huge pressure on the back side of the heart. And the reason it's such a huge pressure is illustrated here. If you actually take, these are trabecular preparations that we took from the atria, and we asked the question, how much force do you actually have to put on these things to stretch these preparations? And you can see what happens with the atria. They stretch no problem, right? You put a little bit of pressure on these guys and a little bit of force, and they're like loose elastic bands. Here's what the ventricle does. It really it's incapable of going beyond, and this is without a pericardium. They just don't stretch. So you jack up the pressure in the backside of the heart. The atria preferentially are getting a massive stretch compared to the ventricles. And that might explain why the atria have adverse remodeling, but the ventricles actually do not. And the other data I'll just pass over. And then the thing I want to point out is that here's an interesting fact that's been demonstrated in a review article by Kovacs. He's actually gone through the literature, and what he's been able to identify is that the wedge pressures, and that's essentially measuring the pressure on the back side of the heart on the left side, goes up to about 20 to 25 millimeters with intermediate levels of exercise. But the thing is, the pressures actually go up much more in older people, over 50, than it does in younger people. And that might actually explain the connection between exercise and AF right, in the elderly population, and in any population for that matter, right, because they're going to respond regardless of whether they have cardiovascular disease or exercise. Their back pressures on the heart are going to go up, and that's probably related to diastolic filling. So I'm just going to end. I'm almost done, Yoram. I don't want to keep you too much longer. But here's some future directions that we've actually performed or engaged in. First is, are these effects reversible, right? Because this is only 6 weeks, and so it's possible that there's actually, that the adverse remodeling that we picked up in terms of collagen deposition is actually reversible, and the answer is no. Right? If you actually wait 6 weeks later, and we're just getting animals now that are 12 weeks later, I don't have the answer to that yet, but certainly after 6 weeks of deconditioning, and their heart rates actually have gone back up. So this is the level in this control group. The heart rates actually went back up, but did not go back to the levels that we actually measured in the sedentaries. And so there's some reverse remodeling that occurs with heart rate, but not the collagen deposition. And the P wave duration also did not reverse, right? So they still have prolonged P wave durations. So there's no reverse modeling of the atria in terms of their structural changes that actually occur.
This is how long after you stopped the exercise?
6 weeks.
So you exercise for 6 weeks?
Right.
And then you look after 6 weeks?
Right. Yeah. And we'll come to the limitations, right, about why it is we get this so quickly, we think, in the mice. So one of the criticisms we always get, or we got when we tried to publish our paper was, well, you're swimming, you're forcing these animals, you're stressing the Jesus out of them, right? And so, it's a stress response. And that's certainly a valid criticism. And so we developed a freewheel running model, plus and minus resistance. And I'll show you what that means. These mice actually run in the course of about 10 days. They run about 60 to 70 kilometers. Okay? So they go quite a distance. And we've measured their work rates, because what we can do is actually measure their O2 consumption. So we do it exactly the same way that you can do it in humans. And so this is where the baseline has actually been removed. And this is just the change in oxygen consumption that goes along with a mouse jumping on a freewheel. And this is a freewheel with resistance. And here's where the resistance was actually engaged. And you can see how much more work the mouse is actually willing to do, about 40% more work. And that's actually illustrated here. If you look at over the course of a night, if you ask the question, how much oxygen did they consume as a result of their exercise, because we've taken off the baseline, here you see with the resistance wheel, we can make them run quite a bit harder, right? We make them do quite a bit more work. And this just shows you the distribution of times when they're actually running at these different levels. So when they've got the resistance wheel engaged, they spend quite a bit more time doing quite a bit more work. And so here's what happens. It's pretty remarkable, actually. So I showed you before swim exercise, freewheel running, right, without resistance, no fibrosis, right? They're virtually the same as the controls. And with freewheel running, there's not as much fibrosis as we got with the swim, but they actually do have statistical increases in the amount of fibrosis. So this is telling us, right, that the amount of work, right, how strenuous the work actually is clearly a factor in this. And by the way, the freewheel runners are doing about 5 times the amount of work per week that our swim exercise mice are doing, right? But the swim exercise, when they're swimming, they're burning way more O2 actually than these guys on the resistance wheel. So it's this kind of spectrum, right, of inputs that are determining whether or not you get this adverse remodeling. It's probably a combination of the total amount of work, but it's also the intensity of the work, which may relate back to how much the back pressure goes up in the heart. This just shows you the percent tissue collagen as a function of the distance run. And you can see here that there's no dependence in the freewheel runners without resistance, but there's a clear dependence. And if you now do this in terms of oxygen consumption, there's a very clear dependence of the amount of collagen that gets deposited in terms of the amount of work that they've actually done, right, over the course of the night every time they're running. Did I go in the wrong direction? No. Okay, and the last thing we've done is we've started to knock out TNF-alpha in different tissues, right, different cell types. And our first pass at this has been to knock it out in the myocardium. And to our dismay, actually, and we're a little surprised. When we knock it out in the cardiomyocytes, we don't rescue the phenotype. Okay, so we're now in the process of actually generating Cree knockouts, and we're really going after the endothelial cells because there's very clear evidence recently that much of the hypertrophic signaling that's actually occurring in the heart generally is actually, certainly through RAF, is actually going through the endothelial cells. So that's our sort of first line. Regardless, we'll actually knock it out in the immune cells. We've already started those experiments as well as in fibroblasts and see which cell type is actually doing the transduction. So this is my last slide, so I didn't do too badly, maybe skimmed over a few too many things. So there's lots of questions that we still need to answer, right? But we think that one of the reasons that you might argue the mouse is not a good model, right? One of it is enforced exercise, but we address that with freewheel running, right? They love to exercise, at least the CD1s. And that's well described in the literature. In the 1960s, there's actually a paper we rediscovered that where they actually had mice on a treadmill. And the mice would actually turn the treadmill on and off on their own to actually do running. It's actually remarkable. And we sort of rediscovered that in our studies when we were trying to do these manipulations. And my postdoc came up with this great idea, and then we found this, like often happens in science, you realize someone's already done it and done it way better than we would have ever done it. But mice are addicted, some of them, right? Some strains are addicted to exercise, CD1s in particular, and they're well described in the physiology literature. They're almost always the ones that are used. So one thing I want to draw your attention to, mouse heart rates we know are 10 times that of the human. Now, they actually are capable of increasing their level of activity to about, with vigorous exercise and swimming, to about 4 times the MET. Humans go to 10 to 15, no problem, right? So there's a big disconnect. As hard as we try to make the mice work, we can't come close to vigorous exercise in humans. But here's the thing that you have to keep in mind. Mice have got 10 times the heart rate, and they've got a limitation on how much they can actually increase their cardiac output. You can increase your cardiac output by jacking up your heart rate by 4 times the baseline rate. The mouse increases his heart rate by about 20 to 30%. That's it, right? So the only way that they can increase their cardiac output essentially is the Frank Starling mechanism, which is what I was talking about before. The backside loading becomes much more important, we think, in the mouse compared to the human, and probably explains the limitation of how much work we can actually get out of these mice, right? But despite that, in 6 weeks, we get, very marked adverse remodeling that actually takes place. The basal metabolism, by the way, of the mouse is 10 times that of the human. And this is well described, right? And this follows Kleber's law. I don't know if you know what that is, but the scaling, right, of the different species. And this pretty much matches up quite well to Kleber's law, right? But again, I draw your attention to the fact that our mice can't even come close to what a human can do, right? And part of that's obviously, a human drives himself or herself, right? A mouse, we're trying to convince them that they should maybe do a bit more. I mean, they're not competing in the Olympics or anything, right? So they clearly have a lack of motivation possibly in all of this. And maybe we won't get into this, but we're very interested right now, though, in trying to understand sort of the complexities of, how much work input is required and try to nail that down further. And the last thing I'll mention is that I've got a Ph.D. student now who's working on interval training, right? Because the big thing in the exercise field is how do you get bang for your buck, right? Everybody doesn't want to spend 20 hours a week, right, conditioning themselves. And it's known, if you bring your target heart rates, right, up to 80–90% of your max, right, you get much more bang for your buck. And that's been shown many, many, many times, right? You'll get conditioning within a week, right, versus a month if you actually use interval training. So we're very interested in seeing what interval training actually does in our mice. And what we do here is we actually swim them to exhaustion. Maybe, I don't think we put it that way to our animal care committee, but we're actually in the process of trying to understand whether interval training might actually impact quite negatively in terms of atrial remodeling. And with that, I thank you for your attention, and I guess I pretty much acknowledged most of the people already, but these two guys were amazing to work with. They're actually, this guy's just taking a job now in industry, a big loss to academia in my opinion. And this guy is actually trying to get himself into medical school, so I'll tell him about Wash U maybe when I get back. So I'm not sure he's got $50 billion a year to pay, but Canadian dollars. Okay, thanks for your attention. Yeah. Yes.
Do you think some of the observations you made here in the exercise where you have physiologic hypertension and stretch could be applied to, just the general population in terms of the relationship between hypertension and AF and that possibly the same mechanisms? And I'm thinking that maybe patients who have hypertension, besides being treated for their hypertension, which isn't always successful, if you were to give them some sort of, TNF alpha blocker or something, intervene in the mechanism to prevent this fibrosis. Because it seems to be in every model I've ever seen where fibrosis is a player in AF, there's no reversing of fibrosis.
Yeah.
Once you got it, you're stuck with it.
Right, and the only example I know of where fibrosis gets reversed in a cardiac condition is the trout. The trout every winter actually goes through massive fibrosis in the heart. I don't know why. Every spring it resolves, right, in fish. Humans and every animal model, that's right. Once you get fibrosis, it's a one-way street. We suspect that that's going to be, of course, the same thing in our model. Coming back to your question of hypertension, a couple things. So a couple things I don't know, and that is, how high are the back pressures on the heart in hypertensive patients? It's certainly largely fluid driven, right? That's certainly the kidney is very much a player in hypertension. I have my own bias right now. My guys argue against me all the time, and this is why I want to go after the endothelial cells. Because I think, somehow the atria, it's like maybe a double hit kind of thing, right? If you get hypertension combined with stretch, right? Now you actually
some diastolic dysfunction.
Well, diastolic dysfunction is certainly a big part of it, because as soon as you get
higher
diastolic
pressures.
dysfunction, the back pressure builds, right?
Right.
Just to be able to fill the
Right.
heart up. And I think that's kind of the common mechanisms when the title I submitted for the talk was, that this was a potential model for all forms of AF. And that's actually the connection, right? The notion that, it's really a common atrial stretch. And we know, valve disease is a big driver, right, for AF. Hypertension's a big one. And I think there are, and I don't really know the numbers, I should, in terms of how high the left filling pressures are in hypertension. But certainly after you've developed hypertrophy of the heart, it's not hard for me to imagine that those have gone up, right, because of diastolic dysfunction. Right? But you've hit on what I think is the common mechanism between exercise-induced and other forms of AF, right?
And the other question I had is you were doing, I guess, dynamic recordings of the EKGs in these animals for long periods of time.
Correct.
Did you ever do any heart rate variability analysis?
Oh, yeah. So I skimmed over that, actually. We did a lot of that. Turns out heart rate variability goes up quite a bit. Actually,
And I was wondering, at night, because, one of the risk factors you showed was, eating, sleeping, and stuff like that, if any of these animals had any arrhythmias at night.
Yeah, so the answer to that is no, and I mentioned that we never picked up any triggers, right? And they clearly never went into AF. However, it's an interesting point that you're making or drawing attention to, and that is, if you look at heart rate variability in a mouse, right, almost all of it is at night, right? If you actually look at heart rate variability during the day, if you analyze ECG variability during the day, you get very, very little heart rate variability. It's all happening at night, right? It's happening when the heart rate actually drops down. And it's kind of like hitting a ceiling, right? Like if you push the heart rate up towards the ceiling, there's no room really for fluctuations. At night, they drop down by a good 15%. And at that point, you pick up very, very nice heart rate variability. In fact, all the early studies, we looked at heart rate variability 10 years ago in the mice, and I concluded it's all bullshit. It's all crap, right? And the reason was because we didn't do it the way we're doing it now with telemetry, right? At night, and that's where you pick it up. Very clear, exercise increases dramatically the heart rate variability. And the beauty of it actually is, just like in the human, you can knock out the high frequency component with atropine, and you can knock out the low frequency component with propranolol. It's just like the human. Yeah, it's pretty interesting.
So with the atria of the mouse are so small, You probably get very good optical mapping that covers the entire atria. Do you see consistent triggers coming from the pulmonary vein?
No, no, the answer is no. We've actually tried, like we've isolated the atria along with the pulmonary veins and looked really hard to see if we could pick up anything. Even with isoproterenol, we tried all kinds of stuff. The answer is no. We do actually, we've actually got the model for the CAV 1.3, the 1D calcium channel knockout. They get triggers in the veins, and it's interesting because you can induce AF in those animals. Actually, that's been published already anyway, but they do get triggers. So unfortunately, the student that finished that project went on and is in medical school now. So we haven't followed up on that.
It'd be nice if if you could characterize more
Yeah.
the actual mechanism of the atrial fibrillation in this particular.
Yeah, so that's why I threw those slides in, right? Because I wanted to get a little bit of feedback. I skimmed over it, the notion of the complexity that we're picking up. But the nice thing about it is that despite the fact that those atria are so small, right, we still get the heart block, we still get the migrating rotors that come and go. So it's very much like real atrial fibrillation, I think, right? But not all of them, right? Some of them, when they induce, they're clearly one dominant rotor that anchors in the right or the left atrium, right? But you see the conduction delay across the isthmus, et cetera, right? Across the nodal region, et cetera, yeah. Sure.
So I have a question regarding along the same lines. So when you start exercising these mice, do they start with paroxysmal and then gradually progress towards
No.
persistent? And how does the mechanism complexity change after you stop exercise?
Yeah, so I can't answer the second one, although we've done it, I just don't know off the top of my head. In terms of paroxysmal versus persistent, for us there's no such thing. We had to induce, right? And so they, as I pointed out, when we did telemetry on these guys, even throughout the exercise periods and thereafter, we never picked up any AF events. We never even picked up any PACs or any evidence of, yeah. So, I mean, one of the things, of course, now that we have these resistance wheels, we can let them run for a month or 5 months or whatever, right? We're doing that now. We're just letting them run every day, and they love it, right? We've actually enhanced their lifestyle considerably. The only thing they need now is color television, right? That they can watch television as they're running.
Maybe we should have a mouse Olympics.
Yeah, sure. We got some pretty well-trained mice, I'm telling you. But there's a paper actually about 3 or 4 years ago from a group that actually was doing exercise physiology, and they noticed, and we know that too, we noticed it too, that some mice, the CD1s are what am I trying to say? Heterogeneous genetic population, right? They're not a homogeneous population. So there's some mice that really run a lot more than the other mice. So there's a study that's been done where they actually took the hard runners, right, male and female, and they bred them together. And you should dig out this paper. It's pretty cool. They produce these super mice that just run like crazy, and they actually end up with some pathology, actually. Yeah.
So I guess building on the same concepts about fibrosis, fascinating part of it. So I guess once the fibrosis is developed, there's no going back. And I'm just sort of thinking about the translational aspect of it, and I'm sure your sort of knockout, different DNF knockouts would sort of sort that out a little bit. But I guess the only real anti-inflammatory we use to prevent sort of disease is aspirin.
Yeah.
And I guess we've sort of looked at the TNF sort of pathway, but in the role of any other anti-inflammatory agents in preventing, I don't know if there's any human data on aspirin and
Right.
the risk of their failure.
So there is a paper or two where they've suggested taking like steroids and things of that type. I think it's been mainly just suggested. I don't know of anyone that's actually ever put individuals on that. But certainly a logical thing to try would be anti-inflammatories. And we've actually talked about doing those. It's just a matter of priority. We've never gotten ourselves around to doing those studies. I think it's a great suggestion because it seems to me that would be a relatively cheap way, right, to slow, even if we found it slowed the process, right, that might be actually quite useful, right, for athletes. And we know, right, that inflammatory factors appear in the blood, like marathon runners, I mentioned that with, troponin I being released, but also a number of inflammatory factors appear, right, in the blood with marathon runners as well as other athletes. So, it is an inflammatory, exercise is an inflammatory promoting activity, right, for sure, right? And so the extent, though, to which that's actually contributing directly to our atrial remodeling, we don't really know. We would prefer to think that a lot of this is actually local, but that doesn't mean that the anti-inflammatories would not be useful under those conditions either, right? Yep.
You make a really interesting point about how athletes and some of them will go on to get atrial fibrillation, particularly at a young age, and some people don't.
Right.
And there's bits and bobs of literature about this being sort of a channelopathy in some.
Sure. Yeah, and there's a number of candidate genes that have already been identified. As
What's your feeling on that concept, and do you have any sort of way in which you can use this model to study calcium?
Yeah. So I don't. But coming to the point about, are there genetic predispositions, right? I think that there almost certainly are. I just know, like in my own family, just as an example, right, we probably are considered defective in many ways, right? But I have 3 cousins, 2 of them are in Holland actually, who are avid cyclists, right, with AF. So I got 3 cousins, first cousin. On my father's side, right, almost everyone has a bradycardia, right? And that's kind of something that is connected actually, right? Six-sided syndrome is something that almost goes hand in hand, with chicken and egg comes to mind here. But nevertheless, there's a tendency, right? The larger your vulnerability period, right, between beats, is the more likely you are for a trigger to potentially impact on the normal electrical patterning of the heart. So, but that's not, there's probably, many factors, right? Sodium channel mutations have now been identified, beta subunits, the whole thing. So yeah, once the more of that information gets out there, you can begin to screen individuals that are picking up AF at the age of 40, et cetera, right? So, and maybe in time we'll start to understand the extent to which there's a genetic vulnerability, right? Not just, it's like a double hit thing, right? If you're vulnerable already and now you engage in exercise, boom, right, you're going to be more vulnerable. And that's probably true in the cardiovascular disease patients as well, right? Some get AF, some seem to actually do quite well and not develop AF.
And also your bradycardia point is very interesting because with your veterinary background, horses have a very low heart rate.
Yeah.
But they have a very high incidence of atrial fibrillation, very easy to use them as models.
They do, and in fact, there's an article that we found in the veterinary literature, had a horse on a treadmill, right? Pulmonary back wedge pressures, 70 millimeters of mercury. And I don't know if you are people who go to the races, right? But Lasix is routinely given to horses after a race, and the reason is because they get pulmonary edema, and blood comes basically, out their nostrils at the end of every race. And the idea there, in all likelihood, those animals have been bred to run, right? They've been bred to just put out like massive cardiac output. So 70 millimeters of mercury on the backside, right? Pulmonary hypertension is massive in those animals, right? And they've actually done like lung studies, and there's bursting of capillaries. Major damage happens in a horse race. After every race for that horse, he's got several days where he's got to repair his lungs. Yeah, and AF, as you point out, is very, very common in racehorses. So I think that it all kind of makes sense with respects to what we've observed in the mice, I would say. Yeah.
Any other questions or comments? Thank you very much for a great talk.